Hello everyone, and welcome to today's live broadcast, Quality by Design, Development of a Purity Method using UPLC and Mass Detection. I'm Gordon Carlson of Waters, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Waters. For more than 50 years, Waters Corporation has developed innovative analytical science solutions to support customer discoveries, operations, performance, and regulatory compliance. For more information, please visit www.waters.com. A few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can at the end of the presentation. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button or submit your problem through the green Q&A button on the lower left. I would now like to introduce today's speakers. Alexander Schmidt and Mijo Stanic are the founders and managing directors of Chromicent GmbH, a pharmaceutical service provider based in Berlin, Germany. Their company specializes in the development of efficient and robust HPLC and UPLC methods. Alexander Schmidt is an expert in the development and validation of analytical methods, applying the very latest quality by design approaches. He's the author of more than 50 publications and scientific posters, as well as being a guest lecturer at the Beuth University in Berlin. Mijo Stanic is also well published and an expert in analytical development within the framework of quality by design. He specializes in the development of LC-MS methods. Today we are very fortunate that Alexander and Mijo have taken the time to describe their quality by design approach. We hope that you will enjoy their presentation.
Yes, thank you very much for the introduction. So let us start with our presentation. We call it We Are Designing Analytical Methods. So the agenda, what we would like to speak today, who we are, Chromicent, the company Chromicent. Then we would like to compare traditional versus systematic strategies for method development. The last point, Mijo will show us a case study, quality by design and analytical method development, a case study. So who we are. I'm Alexander Schmidt. Mijo is sitting next to me, and we are the founder of Chromicent. We have together more than 35 years experiments in the pharmaceutical industry in developing and validation of UPLC and UHPLC methods, in quality by design, in analytical instrument qualification, GMP, and productivity and efficiency increase. We published more than 50 publications and presentations on international congresses, and we are very often invited speakers at conferences. Our company, Chromicent, is a pharmaceutical service provider, and we are offering method development and quality by design framework, forced degradation studies for method development and impurity profiling, method validation, method transfer, as well as consulting, training, and GMP auditing. Our company is based in Berlin, Germany, in a technology park which is called Adlershof. Adlershof is one of the biggest technology parks in Europe, and we are here we are integrated in network activities which are called Analytic City Adlershof. We are also a reference customer and training facility for the Waters Corporation. We have more than 440 square meters lab space, and by the end of the year we will have a staff of 12. The equipment which we are using are aligned HPLC systems with different kinds of detections, UPLC, Accuracy Classic, H-class and I-class systems, also with different kinds of detectors, inclusive of single and tandem MS detectors, We also have an Acuity UPC square, which is an ultra-performance FFC system from Waters, which is also connected to MS detection. And we have automatic sample preparation robot stations and the solution testers from the Zotac Corporation. The analytes we are currently developing methods for are mainly small molecules, a lot of generics inclusive, antibiotics and hormones, anabolic steroids, stimulants, pesticides, natural components, and carbohydrates. Now let us make a comparison between traditional and versus systematic strategies in method development. This is mainly done by trial and error, by varying one factor at a time. The problems which could occur if you are using such a traditional strategy is that additional or missing peaks you see very late in the development process, and trying to test quality in a method is always the wrong way. So this result is very time-consuming, and you have no understanding of the influential key factors. I would like to give you an example for this. If you are, here you have the two parameters x and y, and in a one factor at a time approach, you fix one factor, that is in this case y, and varying the factor x, until you find a working point. Let's assume that the working point would be here, the black dot. In the next step, you would fix the factor x and varying the factor y until you find another working point. But what really happens is that the robust region is next to the working point you have selected by the one factor at a time approach, and therefore you wouldn't find very robust methods. So what could happen if you have not a non-robust method? We would like to show you here in some chromatograms we received from the Molnar Institute. The first chromatogram looks very nice. You have a well separation between all the peaks. The peak symmetry is good, but from day to day or from system to system, you get variations. And you can see that especially in the green box, that something happened. And the question is, what happened? And you can see it here in the last chromatogram. One peak was in the second, in the last peak, in the second peak of the first chromatogram, then in the first peak of the second chromatogram, and finally it moved out of the peaks, and now we have the separation of these three peaks. And if you find that out very late in your development process, maybe at the validation, or maybe when the product is already on the market, you are in deep trouble with this kind of method. Another traditional strategy in method development is to adapt already existing methods, maybe from the literature or published in Pharmacopoeias. Here's a very nice example of an official method which was adapted from the current version of the European Pharmacopeia. It is the official method for Ebastin, and you can see here that the runtime is unbelievable, 160 minutes. The peak width for the Ebastin peak is 8 minutes, and the retention factor is between 0.25 and 70. That means you have the first peaks eluted with the solvent, and the last peak it takes forever until it comes from the column. in comparison to the traditional approach, there is a systematic method development approach, for example, by using statistical software tools. In that approach, we are using experimental design plans, and in a full or in a fractional factorial design, you perform a couple of experiments to find the best method. Typical software packages for this is the Fusion software and Design Expert. Another systematic LC method development approach is by using modeling software tools. That is a very smart way in which you perform only some center or corner experiments, and the software tools can then calculate all the chromatograms which are within these corners. Typical example are the DryLab software, Chromicent, and from ACD, the LC simulator. Finally, and that is the approach we perform here at Chromicent, is the quality by design approach, which was implemented some years ago also in the ICH guidelines Q8. It starts with the definition of method goals, then a risk assessment, the design of experiments to define the critical quality attributes, and in a screening and optimization phase, you select the best parameter. From all these, you build and design space, select the working point, make a verification, and then start with the validation and robustness study. The final step is to implement a control strategy that could be in system suitability test to make sure that the method is always robust and working in your lab. Mijo is giving us now in a study how we perform such a method development in our lab. Mijo?
So first again, the initial situation, like we have seen before. The official HPLC method for Sebastian, published in the European Pharmacopeia, with unbelievable 100 minutes. So first we have to decide what are our goals. The first is we wanted to use UPLC technology to be much more faster. And in the second part, we want to use for the screening part the statistical software Fusion, and for the optimization part, the modeling software DryLab. So again, the slide with all these 5 steps we implemented in our approach. First, define method goals. What we want to have is a separation of all components with a resolution higher 2.0. Minimum analysis time should be lower than 10 minutes. The K-star values for the API and the impurities between 2 and 10. And what we want to have is a design space that we can visualize, that we can touch. So step 2. Risk assessment, where we have defined all critical parameters that are the columns, the gradient time, the temperature, ternary composition of the eluent, pH of the eluent, and start percent B and end percent B. Next step, design of experiments, where we screen for columns. There are more than 1,000 different columns on the market, and on the Waters homepage we can find these graph with many different columns. And on the selectivity charts you can see the hydrophobicity against the selectivity, and all these blue dots are columns. And what we wanted to do is to use UPLC technique, and therefore we need sub-2 micro columns, and we use Waters UPLC columns. All these gray and red dots are available columns from Waters for UPLC on the market, and we decided to take these 4 columns. Why? Why we have used these 4 columns? First, we used the BH C18 column. That's a very robust column, and try to find orthogonal columns that are available on the market. For this, we used a BH phenyl column with a very similar selectivity, but very different hydrophobicity. And very orthogonal to the phenyl column is the HSSC18SB column with the same hydrophobicity, but a different selectivity. And because we use the 2 BH columns, that's why we choose the HSS-T3 column to have 2 BH columns and 2 HSS columns. So all these experiments were run by the H class. Why the H class? Because you have the chance to take to use 9 different solvents, like 3 organic solvents and 6 different pHs. You can use with the column manager up to 6 different columns with different temperature zones, and we use the PDA detector and the QDA single MS detector for peak tracking. So, implementing the benefits of 2 software tools. For the screening part, we have used the Fusion software, where the software is automatically implementing all the methods in Mpower, writing all your instrument methods, your method sets, your sample set, and so on. With this software, you can use all your 6 different columns and 9 different organic solvents automatically. And with the software, you can use a brighter parameter range. So, like for the pH, where you can use from pH 2 to 14, for example. And for the optimization part, we used DryLab, where we have to run only few runs are necessary. The variation of flow rate, start %B and end %B, is possible to run without experiments within the software. With DryLab, we can receive predicted chromatograms. For example, the software is showing you how the chromatogram should like. And the robustness testing we can make in silico, where also no runs are not needed to do. So, next step, screening power with Fusion. First, we have to create a design. We have fixed 3 parameters, the flow rate, the start %B, and the end %B, because the variation is later possible with DryLab without performing some runs. And we have some variable parameters where we used acetonitrile, methanol, isopropanol, and for pH, we used all the range from 2 to 7. The temperature is 35 to 70 degrees. The gradient time 5 to 10 minutes, and the 4 different columns with the same parameters like we have shown before. So next part, building the design of experiments. The software is now creating your completely sample set, where we have 78 different chromatograms or runs. After implementing it in software, we have to run your experiments, and what you get is 4 different design spaces, where we have to choose as our parameters all our critical quality attributes, like the resolution between all our peaks, the number of peaks, the symmetry factor, or the retention time of your last peak. And everywhere we have met our goals, the area should be white. So you can see with the HSSC18SB column, there's no And there's no possibility to get a nice chromatogram with the goals we have decided. And with the BHC18 column, this column has the biggest white area. So we should take the BHC18 column to the optimization part. So, selection of parameters for the optimization. We have fixed again the flow rate, the start percent %B, the end percent %B, and this time normally the column, the BHC18. And the variable parameters are now acetonitrile and isopropanol. The pH should be is tested with 5.8 and 6.8. The temperature is again from 35 degrees to 70 degrees, and the gradient time is a little bit shorter with 3 to 9 minutes. So for the optimization phase, the design of experiments were done with dry lab, where we've chosen 2 different temperatures, 2 different gradient times, and 3 different ternary composition of eluent B with acetonitrile and isopropanol. And on the first layer, we can see our design space with 100% acetonitrile, where we can see the gradient time against the temperature. And let me explain you a little bit the color code. The red color shows a resolution higher than 2.0, and the nearly black area shows you a resolution with 0. And all the colors between red and black are the resolution between 0 and 2.0. And here we can see with 100% acetonitrile, we don't receive a resolution with higher than 2.0. But when we increase the amount of isopropanol to 30%, we get with a higher temperature a red area where where our goals were met with a resolution higher with 2.0. But with 60% isopropanol, we get an almost red area. So this shows us that everywhere we can choose with the temperature or the gradient time, we get a resolution higher than 2.0. But when we combine all the 3 layers, we get the cube. But in this cube, we see all the other different colors, but by resetting the parameters, we receive a big red area, and that is our design space. Now we can choose our working point somewhere in the middle. We can choose somewhere in the middle. So next step would be to test the influence of the pH. This time gradient time against pH from 5.8 to 6.8. And what we get is again a design space, a red area, where all our components have a resolution higher than 2.0, but the black area is this time a resolution 1.5. That means all the colors between black and red are the resolution between 1.5 and 2.0, and that's the edge of the failure. That means if you choose your working point on the edge of a failure, you are close to the that your chromatogram is not working anymore. So what we have done is a verification of the model with real experiments, where we have chosen our working point and run the experiment, and 6 different verification points around our working point. You can see the dry lab predicted retention time against the experimental retention time. But there are too many numbers to compare, so what we have done to make a graph with the predicted retention time against the experimental retention times, and what we get is a correlation coefficient of 0.9996. That means that our predicted design space is true and very good. The next step would be method validation. Alexander?
Yeah, the method validation study was performed in accordance to the ICH guideline Q2, of course, and with the steps specificity, linearity, accuracy, precision, and the last step in a method validation study, you know it, is the method robustness. We always recommend to do the robustness study before you perform the validation study, just in case that you find some issues which make it necessary to move your working point in some directions. So in this case, we had optimized 6 parameters, and in a full factorial design with 3 levels, we had from 3 to the power of 6. That means 729 experiments. I think nobody is performing this in a method validation study, but we used the previous design space, our model, for the robustness testing. And by using the DryLab software, we can do this in silico. That is a very nice feature in the new DryLab version 4.0 to perform the robustness testing in silico. The results of these 729 experiments, you can see here only the first 10 runs and the last 10 runs. And in the last column, you can see the critical resolution between of all critical peak pairs, and there is the resolution is always higher than 2.0 for all experiments. That means the method is very robust. Last step in our development step is or strategy is to implement a control strategy. Because the method is so robust, we choose the resolution of the critical peak pair, which we identified as impurity C and D as a system suitability parameter, and that was all for our control strategy. And here now you can see the final UPLC method, which we developed by using Fusion for the screening and DryLab for the optimization. You can see a very nice separation of all impurity peaks from the API, the Abestin, and the runtime is only 4 minutes. And here, once again, the comparison of the final method against the official UPLC method. The official method from the European Pharmacopeia, 160 minutes, and our new developed method, 4 minutes. So conclusion, all previous defined method goals were met. We have achieved baseline separation of all components of interest with a resolution higher than 2.0. The retention factor values for Abestin and impurities are between 2.2 and 4.3. That is perfect, fits into the 1 to 20 value range. The design space is visualized, and the analysis time is only 4 minutes. That's an impressive 40-fold increase in the productivity in comparison to the method published in the European Pharmacopeia. And it allows testing for more than 360 samples per day in comparison to 8 samples with the old method. Yeah, that's it. Thank you very much for your interest. And if you have any questions, please let us know.
Thank you, Alexander and Mijo, for that informative presentation. The presentation and PDF files of the presentation will be sent to all attendees. And now I would like to just remind our audience how to submit questions. You can submit by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Our first question: which type of regression model do you use to explore and define the design space?
I don't really know, because we used only the Fusion version for non-experts. So Fusion gives you basic options, so it takes statistical designs for you. So you don't have to choose what kind of model. That's only in the expert version. So I hope I've answered the question. For DryLab, there is no model. There are only calculations were done.
Next question. If you are using an MS detector, is resolution still a critical parameter? The MS data will provide masses for co-eluding peaks.
MS is only for peak tracking, not for the resolution, so we can see where the peaks are moving.
To explore the space for the composition of the eluent in the three components, do you use D-optimal design?
No. There's only, I think in statistical, it's a D-optimal design. And like I said before, I don't really know what Fusion is doing behind. So that was only for the non-expert version we used. And DryLab is not using the optimal designs.
Have you used the PQRI database for column selection or screening?
Yes, we have, but you will get just a number for the comparison of all these columns, and we prefer a graphic comparison, and that's the reason why we used the column selectivity chart on the Waters web page. We think it's just easier to see and to handle.
Could you please comment on the matrix effect using this type of method development?
I don't know if I really understand this question. There was no matrix effect because it was only the API and the impurities.
Can separations using mixed mode HPLC columns be modeled with this software?
No, as far as I know, DryLab software can only make calculations with reversed phase, in the reversed phase mode, and normal phase, but not in combination of that, as far as I know.
While designing design of experiment methods, when do you select partial factorial and when do we select full factorial?
Easy to answer. When 4 experiments are full factorial, so we do full factorial. It depends how much parameters you have. The more parameters you have, you need much more experiments to make a full factorial design. So full factorial only makes sense for a number of parameters.
When you develop a method in Fusion, how do you track unknown peaks of interest from degradation? For example, trying to get a stability indication method.
We have also looked for unknown, but the impurities from the degradation are mostly these known impurities. But after that, we have also test on forced degradation impurities. So,
how do you perform peak tracking with this software? Is it automated, or do you need to manually assign the peaks?
Peak tracking is manually when we use DryLab, because the software doesn't know how the peak is moving.
Does the software calculate the probability of the model designed for you according to your input and validate it at the end as well?
No, of course not. You have to validate your final method. It's up to you where your working point in your design space is. So the validation study has to be performed by the user after the selection of the working point and verification study.
How is the QDA used for peak tracking and purity analysis?
Yeah, the QDA is very useful in to see where your peaks are moving. So sometimes you have impurities which are very similar to the spectra of the impurities are very similar to the main peak. So if you have to make a peak tracking to see whether the movements or where your peak moves, you need more data than only the UV spectra, and in this case the QDA detector is very useful.
Were the 729 experiments actually simulations, or were they performed experimentally?
They're only simulations because we have verified the working point and 6 different verification points. We showed that our design space, our simulations, are true. So there was no need for do some other experiments for the robustness testing, only to use the software.
So are there any situations where fusion or dry lab should not be used in simulation processes?
At this moment, I don't know any part where they are not usable. So, but we haven't checked everything what's possible.
How long does it take to complete the total process from start to finish?
It depends. Sometimes 3 days, sometimes a week, and inclusive the validation study. In 2 weeks, we are finished with method development and the validation.
We are doing only for developing one method.
How do you deal with unknown mixtures where you might not know all of the components? Or alternatively, do you need to know the composition of a mix to do optimization?
No, there is no need to know the molecules. Of course, it's a help if you know what kind of molecules you have, but the fusion and with dry lab software, you don't need to know what it is. You can separate the mixture without knowing the molecules which are in the mixture.
How did you come up with the range in the design space for the fusion experiments?
The ranges are, you are deciding in the beginning what is your lower and your higher limit, and the slides are showing you have to choose to fix some parameters, and then you have a 2D graph where you can choose your working point, or changing the fixed parameters, for example, pH not at pH 5, then you can change to 6 and see what's happening then in your slide, your 2D graph.
Are there any guidelines to set the relative standard deviation in impurities analysis?
You know, the ICH guidelines, of course, are giving you some advice in impurities and what the specifications are, but I don't know if the relative standard deviation is part of these ICH guidelines. So I would advise check the ICH guideline QA and QC, please.
Could the design space be used to define a method in the MA dossier in order to let the laboratory have more flexibility in the analyses? For example, in the case of a transfer to QC?
Yeah, that's exactly what we are doing. So We have a working point, but also a range, and we put this information in our dossier so that in case of a method transfer to quality control or to a different lab, you can use this information.
Do you have any advice for injecting a sample with high percentage of organic?
In a method development strategy where you start with a very low organic composition, it's a little bit tricky. So at the moment I don't know what could be done without diluting, of course, the sample.
Have you submitted any modeling data to regulatory, such as FDA or EMA, and what was the response?
We submitted our approach, our strategy to some European national regulators, and they accepted it without any questions for the analytical development and validation study. So it was perfect.
Have you tested Hillock columns in the screenings of polar compounds?
No, we haven't.
Is there any software which is able to design the experiments for sample preparation time optimization, like sonication, shaking, etc.?
Yes, we are using Design Expert software for the sample preparation. And also we have an automatic sample preparation robot station to perform all the experiments in our experiment plan.
Do you have some more examples other than Ibacine to support quality by design?
Yeah, sure, of course. So a lot of more, and if you check our names on the internet, at Google, for example, you will find more examples. One was published, for example, in the ACGC. I think it was in the February issue of this year in North America, and in September in Europe. So with a lot of examples published.
How many experiments were run using the Fusion AE software?
For the screening part, we needed 78 different experiments.
When do I choose between Fusion or Dry Lab for method development? Are both necessary?
No, but we wanted to use the benefits of both softwares. There's, in my opinion, no software that's you can say it's the best software. So we use the benefits of one and the benefit of the other software to get much better design or a faster way for QBD.
I have used Dry Lab for LC method development with good agreement between the predicted and actual data. Do you have any experience for GC method development?
No, only LC and SFC.
I'm interested in your comment regarding using the Fusion software to create your Empower methods. Can you describe this process in a little more detail?
Fusion is working with a toolkit in Empower, and all the runs, method sets, sample sets, instrument methods are written from Fusion in the Empower software. So you only have to open the sample set and run the whole sample set. The processing method have to be done by yourself. And then again, implementation of the result set in Fusion again.
Do you have any data on Design Expert software?
Yes, but only for sample preparation.
But these data are not published yet. So it's only for us and for our customers we have made some data.
We're not allowed to publish everything.
You know the impurity molecules, but you don't succeed in separating them. For example, you don't succeed to separate IMP from IMPY, but you can see that they're co-eluting using QDA mass data. Can you use this as an input in dry lab or fusion? And if so, how?
Yes, you can use that in dry lab, this information. Just type in the same retention time for the API and your impurity, and then the dry lab software realize that you have a co-elution of two components.
Does the software work for sample set creation in mass links as well, or only in Power?
I think this question was for fusion software. I don't really know. Ask S-Metrics.
Yeah, you know, as a pharmaceutical service provider, we only use Empower software and not MassLink. So we have no idea if that works, or if fusion software will work also with MassLink software. Sorry.
The fusion that you talk about is only works with Empower. MassLinks is not supported by that. If you don't have any software, can you also apply quality by design approach for your method development?
It's difficult. If you can handle all these peaks and chromatograms, I'm not really sure. It looks like then more like trial and error.
How do you evaluate the method predicted by dry lab?
Yeah, we used a verification study. So dry lab is giving us is showing us the best method or just a method, and so we can make a comparison between the retention time of the prediction and the retention time in an experiment, and so we get a verification if the model is accurate or not. And we just saw it in our slides. The comparison is about 99.9%, so very, very, very good.
Okay, sure. Now, one more question before we close up for the day. Where is the software available?
Dry lab software is available by the Molnar Institute here in Berlin, Germany. The web address is molnar-institute.com, and the fusion software is available from S-Metrics in San Diego, California. And I think the web address is s-metrics.com.
Well, unfortunately, we are out of time. We're going to get back to any unanswered questions later on. We hope that you found Alexander's and Mijo's presentation valuable. If you'd like to learn more about their quality by design approach to LC methods, you can reach them at chromicent.de. There are also a number of scientific downloads on this webinar's registration page. As mentioned before, a PDF of the presentation will be sent to all attendees, and the webcast will be available for on-demand viewing through April of 2015. You'll receive an email from LabRoots alerting you when this webcast will be available for reply. We invite you to forward that announcement to all of your colleagues who may have missed today's live event. See you next time, and goodbye.